This video is to give you a quick overview of what will be going on in the lab for the next 2 days or so as we do our SDS-PAGE and Western blotting practical class. The video is deliberately quite simple. It will provide the basics that we can then build upon, adding complexity as we talk about experimental design and data interpretation at later time points. So in the previous video I explained the whole setup of the experiment. So please watch that video before really continuing with this one, because it'll make it a lot more sense. So in the week leading up to these practical sessions, the people in my lab have prepared the samples that you'll be using in today's experiments and actually tomorrow's too. Basically what we did, we took cells in a dish, transfected them with our different mini-genes or the different control treatments, then left them to incubate for 24 hours. And during this time the cells will have begun to express the proteins encoded by the mini-gene. Next, my team generated a total protein extract from the cells. To do this, they lyse the cells, i.e. burst them open, in a buffer that contains 4 principal components. So firstly, it's got sodium dodecyl sulfate, the SDS from SDS-PAGE. This gives all the proteins a negatively charged that is roughly proportional to the weight of the protein. And this is important as we use electricity to drive our proteins into the gel. So having them negatively charged means that they'll move from the negative to the positive electrode. More on this shortly. So the second component is beta-mercaptoethanol, also known as BME, and this is why your protein solution smells. So BME is a reducing agent. It breaks disulfide bonds, and we use it to break apart any dimers, trimers, or other protein-protein interactions that may exist in your samples. By including it, we'll end up looking at just the monomeric forms of every protein that we care about. Thirdly, we have tris-HCl, which is used to maintain the pH of the solution, and we need this to make sure our proteins will enter the gel. And finally, number 4, we have protease inhibitors. These prevent or at least slow down the degradation of our proteins. In addition, we have a couple of extra things. A blue dye to help us see the proteins at the point of when we're loading them, and a little bit of detergent which actually helps with lysing the cells. Okay, so we're going to hand you ready to go protein solutions which you might have to dilute, and we'll explain that on the day. Your job will be to separate the proteins based on their size using a process called sodium dodecyl sulfide polyacrylamide gel electrophoresis, or SDS-PAGE. And then secondly, you will transfer the proteins from a gel onto a membrane. And then after that, we will probe the membrane with antibodies that are specific to our target proteins that we care about. And then finally, we will detect where those antibodies have bound using fluorescence blot scanning. This will generate an image of the membrane, and we'll use that to interpret the size of our proteins, the relative mass, and which proteins are expressed by which cell types, i.e. from which mini-gene. SDS-PAGE. So day 1 will involve doing the SDS-PAGE part of the experiment. The first thing you will do is prepare the polyacrylamide gels that we'll be using to run the proteins on. This is a 2-step process. First you'll prepare a lower gel, which is also known as a resolving gel, and then you'll prepare an upper gel or stacking gel. To prepare both these gels, we'll assemble a casting cassette consisting of glass plates held vertically within a plastic holder. We'll show you how to do this. And there'll be a small gap between the plates, and that's where the gel is going to go. Before you begin, make sure the glass is thoroughly cleaned, because any fingerprints or dust that you introduce here will carry through right through the experiment, and will end up with smudges or fingerprints on your final membrane. Also check that the casting apparatus doesn't leak, because we don't want liquid gel all over the bench. So usually we'll do this on the day before your experiment, so hopefully we had enough time to do that. When it comes time to prepare the gel, you will follow the protocol in your handbook. There are 5 components to each of the gels. A buffer, tris-HCl, which will maintain the pH. The stacking and the resolving gels each use a different buffer, they are at a different pH, so make sure you use the right one. Secondly, you're going to have an acrylamide bisacrylamide solution. This is the material that will actually form the gel, and it's actually one of the most dangerous chemicals that you routinely use in the lab, so make sure you're very careful with any unpolymerized solution. Then thirdly, there's going to be water, and then we'll use this to dilute the acrylamide to the concentration we need it. The amount of acrylamide, and therefore the amount of water that you use, is dependent on the size of the protein that you want to examine in. Here, because we are looking for proteins that are 40 to 60 kilodaltons, we will use a 10 or 12% gel. You can do the calculation for how much acrylamide you need ahead of the lab. The information is all in your handbook. Once we've added these 3 big volume reagents, and only once everything else is actually ready to go, we will add 2 final reagents that will actually start the gelling process. These are ammonium persulfate and timid. They basically cross-link the acrylamide and turn into a gel. As soon as these are added, you will mix the solution and you'll pour or pipette it between the glass plates up to a line that we will have indicated beforehand. Okay, then we wait. We wait for the first gel to set, and while that's happening, we prepare the solution for our stacking or upper gel. This essentially has all the same ingredients and just a different pH buffer and a lower percentage of acrylamide. Once the lower gel has set and we're happy, then we will pour the upper gel on top of it and insert into that upper gel a comb which will generate the wells that we will then use to load our protein into. Then we wait again for this to set. Each of the setting stages takes about half an hour. During the wait period, we will dilute and prepare our protein samples so that they're all ready to go and at the same concentration. And then, once the stacking gel has set, we will very carefully remove the comb and make sure that the wells have formed properly and that there's no non-polymerized acrylamide within them. At this point, we load the protein, which is actually quite simple. All it involves is carefully pipetting the right volume of our different protein solutions into the wells that we've created. At this point, you need to be careful and take careful note of which order you have loaded the different proteins. Otherwise, we won't be able to interpret our results at the end. Indeed, don't load the gel until you've decided what's the best order in which you should load the proteins, because you won't be able to change this after the fact, and you want to think about what the figure will look like. We'll discuss this on the day and we'll come about a consensus as a class. You'll also load a protein marker into the gel. This is a sample of pre-prepared proteins of known molecular weights that we can use to you can use as a reference point to work out what size our proteins are. So make sure to take note of what the marker proteins are, because sometimes you use different ones. Usually you should load the markers as the first or the last lane of your gel, as it will give you a reference point to help the interpretation later on. Throughout this process, be very careful. Make sure you don't mix one protein with another, or make sure none of the protein flows from one well to the next. Okay, once the gel is totally loaded, you then apply a current. The SDS in our protein solution means that our proteins are negatively charged. So by having a negative electrode at the top, a positive electrode at the bottom, we will drive our proteins down the gel away from the negative toward the positive. The reason that we have a stacking gel and a resolving gel will become clear at this point. All the proteins will enter the stacking gel very quickly. It's a low percentage gel, everything enters fast. But when they hit that transition point between the two gel types, they'll actually stack, hence the stacking name. They'll bunch up, and the net effect will be that all the proteins will actually enter into the lower gel, the resolving gel, at approximately the same time. And then ultimately, once they've separated by size, we'll end up with sharp bands of each protein at its individual size. Okay, running the gel will take probably about an hour and a half, roundabout, so we'll probably have lunchtime. Once the gel is run, our next step is to set up the transfer. So by running the gel, we've separated out our proteins vertically through the gel based on their molecular weights. The bigger proteins will have moved slower, so they will not have traveled as far into the gel as the smaller ones. So now what we're going to do is we're going to use the negative charge on our proteins and an electrical current again to drive the proteins that are now separated from the gel onto a membrane. So the membrane is essentially, it's a type of sort of special paper that has a positive charge on it that makes it very sticky for our negatively charged proteins. It's also not porous like paper. Again, that means that you need to be careful how you handle the membrane. Any fingerprint or dust, again, is going to carry through and we'll have it on our final image. So make sure you're handling it away from the center, away from where the proteins are going to be. So how do we do this part? Well, we make a sandwich. Just after lunch, we should be good at it. At the bottom of the sandwich, we put a sponge, then we put a few pieces of filter paper, then our gel, then the membrane in direct contact with the gel, then some more filter paper, and finally another sponge. The whole gel cassette will then be closed and essentially inserted into a tank and current applied. Simple. All the different parts of the sandwich will be soaked or pre-soaked in transfer buffer, which basically provides a set of ions that the current can run through, and the tank is going to be filled with the same buffer. The biggest point to be aware of while you're assembling the sandwich is that anywhere in the sandwich where there is an air bubble means that no current will flow through that part of the sandwich. In other words, you won't get transfer of your proteins onto the membrane. So therefore, after adding each of the different layers, you're going to roll out any air bubbles to make sure that there's nothing in the system. Okay, so now the current will drive the proteins from the gel to the membrane, and as you'd expect, the smaller proteins will move more readily than the larger ones. And so the amount of time that you put on the transfer is dependent on the size of the protein that you're interested in. In this case, about an hour transfer is going to be appropriate for most of the proteins that we care about. After the transfer is finished, we will disassemble the sandwich, the cassette, and do a quick check to make sure we have successfully transferred our proteins. To do this, we're basically going to use a generic protein dye, Ponceau S, which will stain all the proteins in our membrane a sort of reddy pink colour, and we'll be able to see if we've got any holes or if we had any problems. Assuming everything looks good, we'll be ready to move on. And what we'll do next is we'll put our membrane into a milk or plant protein solution to block. So what this means is that every other part of the membrane is going to get covered by the plant protein. Basically, like I said before, the membrane is a really sticky sort of bit of paper that proteins like to stick to. So if we don't block all other areas prior to probing with our antibodies, then our antibodies will just stick everywhere onto the membrane and we'll just end up with a black mess. The block step can take sort of 30 minutes to an hour, but we might choose to stop here and put our membranes in the fridge overnight depending on the time of the day. Okay, the next day, to finish the experiment, we will need to visualize where our proteins of interest are. To do that, we'll use antibodies that bind specifically to our target proteins, and we will then detect where those antibodies have bound using fluorescently conjugated secondary antibodies. The first step here is to select the right primary antibodies. We have two proteins of interest, so we want antibodies against them. Those are GFP and M. cherry. So use one set of antibodies that bind to GFP. They have been raised in either a mouse or a rabbit. And then you have another set that recognizes M. cherry, which are raised in the other species. So take note at this point whether the mouse antibodies or the rabbit antibodies recognize GFP or M. cherry. Okay, we dilute the primary antibodies down to a concentration that people in my lab have predetermined. Then we apply them to the membrane for about an hour with rocking. During this time, the antibodies should bind very strongly to their targets, but will also bind with lower affinity more weakly to other proteins, including the plant protein. So before the experiments, my lab did some validation experiments to work out the right concentration to maximize the difference between this real signal, the accurate binding, and the noise, the non-specific binding. Okay, once we've done this hour incubation with the primary antibodies, we'll need to wash off any lightly bound antibody that's bound to the wrong place on the membrane. And we'll do this through 5 5 minutes, or 3 5-minute washes in sort of a mild PBS with slight amounts of detergent. Okay, then we'll be ready to probe the membrane with a secondary antibody. So these antibodies have been raised in another species, and they'll bind to, not to the proteins directly, but actually to the primary antibodies. Each of the secondary antibodies carries a fluorescent tag on it, and we'll use the fluorescent tag to visualize where the secondaries are bound and infer from that where the primaries are bound. Okay, because we're using mouse and rabbit primary antibodies, we'll use goat secondary antibodies that have been raised to bind to mouse immunoglobulins with one fluorophore, or goat antibodies that have been raised to bind to rabbit immunoglobulins, and they'll have a different coloured fluorophore on it. Again, you need to make sure that you take note of which wavelength secondary antibodies are binding to which primary antibody. So you've goat anti-mouse with one wavelength, goat anti-rabbit with another. Again, we do the same thing. We incubate our secondary antibodies on the membrane, and they will bind to their targets, which are going to be the mouse or the rabbit primary antibodies. And they bind their target strongly, but they also bind weakly everywhere else. And so then, after an incubation period, we'll then wash off the non-specific binding of the secondary antibodies, and then take our membrane to be scanned with a fluorescent scanner. The two different wavelength antibodies will produce different wavelength light, which we'll detect separately. Okay, so that's it. At the end, we'll be able to interpret which proteins we have in which sample, what size they are, and their relative abundance. And that will allow us to determine what effect each of the different mutations that we're studying will have or has had on our protein expression. The process itself may feel complicated the first time you do it. It's actually relatively straightforward. But throughout the process, make sure you ask lots of questions. There'll be lots of demonstrators available to help you. And the whole process should feel relatively good fun, and ultimately it's a training exercise after all. There's no pressure here. Okay, thanks for watching. I'll see you again.